Hello everyone, and welcome to today's webinar, EMICE Workflows in Every Direction. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leicamicrosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Seth Villarreal, Advanced Workflow Specialist, Nanotechnology, Leica Microsystems. Seth, you may now begin your presentation.
Hello, welcome to this presentation on the EMICE. My name is Seth Villarreal. I'm an Advanced Workflow Specialist for Leica Microsystems. This presentation will be covering the wide variety of workflows that the EMICE supports or is a direct part of. The EMICE is a high-pressure freezer. This enables a number of different ways of working with samples that are all preserved by vitrifying them through high-pressure freezing. In brief, high-pressure freezing, as the name might suggest, uses high pressure under short, couple hundred milliseconds to limit the formation of ice crystals as the sample is very rapidly cooled in order to vitrify the sample and preserve it in this static fashion, allowing subsequent processing in cryo-workflows. Unlike plunge freezing, that tends to work with extremely small volumes, the EMICE works with slightly larger volumes in ranges of 3 to 6 millimeters wide and generally 100 to 300 microns deep. The sample carriers allow much thicker and wider samples to be preserved by vitrification. These volumes ranging several microliters in size are high pressure frozen as controlled through the software and hardware of the EMICE. The software carries out all the internal functions of a freezing process, but a user does need to prepare the sample for the ice to then take over. The EMICE uses a cartridge system. A fully assembled cartridge on the left of this most recent image is a cylinder. There are an upper and lower cylinder halves, as well as a middle plate. These three components serve to hold the sample in X, Y, and Z, preventing it from moving it around, while still allowing it to be high-pressure frozen. In the center of the middle plate, there's a small hole. This is where the sample sits. Generally, samples are enclosed within a small carrier. There are wide varieties of assembling the sample, and I will cover these in brief as it comes up. All the samples are contained within an assembled sandwich that fits within the hole of the middle plate as part of assembling the cartridge. So, here are the workflows that I'll be covering in this presentation. Some of these occur before some during the process of high pressure freezing, and many of them take a high pressure frozen sample and modify it further in order to get the desired data. I'll be starting at the beginning, looking at ways of preparing a sample and collecting data before going to the EM-ICE. The coral life is a workflow that starts with live samples that are imaged on a wide field microscope and then very rapidly transferred to the EM-ICE, allowing preservation of the sample and making use of the light microscopy information in order to capture a particular moment of interest for subsequent analysis by EM. The Thunder Imager Nano is the particular wide field microscope that is part of this workflow. It uses specialized sampling chambers which are dedicated in this workflow in holding the sample. Notice that there is both an in and out port for gas exchange, and you may also be able to see the black middle plate on the bottom of the sampling chamber visible through the transparent top, such that the sample is held in the middle plate, ready to go to the EM-ICE at a moment's notice. This allows imaging of a sample over a period of time, maintaining the sample in appropriate atmosphere and temperature, and once the sample is fully imaged, such that either a biological process is completed or a particular transient biological process is located, the sample can be transferred to the EM-ICE, with this sampling chamber allowing the very rapid assembly of a complete cartridge, such that it can be frozen within 5 seconds of the transfer off the Thunder Imager Nano. In this process, the sampling chamber is picked up out of the microscope, placed on the work area of the ice, and then in 3 simple motions, the first sliding the sampling chamber up to disconnect the middle plate from the chamber, followed by closing a specially designed lever arm to seal the sample within the middle plate before closing the lid of the EM-ICE, completing the overall cartridge assembly. To show this at speed, here is a brief video. The sampling chamber placed on top of the EM-ICE, middle plate removed, covering the sample and completing the cartridge. 5 seconds from imaging to frozen. This process allows live cell imaging, as well as benefiting from the blur removal of the Thunder software. Then the sample, after high pressure freezing, could, for example, undergo free substitution and resin embedding. Subsequent ultramicrotomy and TEM imaging can make use of the coordinates and visual information from light microscopy in order to identify the particular cell and make use of the TEM ultrastructural information in combination as a correlative workflow to examine points in time with a high degree of accuracy. One separate note on this process: high pressure freezing cannot make use of glass cover slips as the glass is too delicate. The light microscope relies upon a sapphire-corrected objective, as the sapphire discs are capable of surviving the high pressure freezing process. The CorrelLife workflow is designed in totality to make use of all these individual capabilities into an overall workflow. Coming back to this overview slide, there are capabilities of the ice that happen during the freezing process. I'll be starting with light stimulation. Light stimulation allows the use of optogenetics and other similar workflows directly within the freezing chamber of the EM ice. The ice freezing chamber has a small sapphire window allowing illumination by a light module as controlled by the software of the EM ice. Various single wavelength modules with programmable intensity and timing allows precise stimulation of a sample during an experiment. Unlike the metal carriers that are very common, stimulating a sample via light requires a transparent surface. Just like the sapphire discs being a requirement in the CorrelLife workflow, they remain a requirement in light stimulation. The sample must go through the EM ice with a visually transparent cartridge and the sapphire disc to allow the light to reach the sample. The sapphire discs are stacked with spacers to create the correct height as supported by the middle plate, and then the cartridge halves are transparent to allow the light passing through the freezing chamber to reach the sample. With the process of light stimulation, there can be a period of equilibration in dark phase, and then a defined set of pulses in order to achieve particular targets of stimulation, as well as time intervals after stimulation to look at different points in time in a triggered biological process. In this example, vesicle recovery after stimulation shows different time points at membrane recovery after vesicle release and fusion. There is one other workflow I should touch on that occurs during the process of high pressure freezing, and this is electrical stimulation. Like optical stimulation, applying an electric field to a sample, especially neurons, can allow examination of vesicle activity or other functions of a cell in order to look at different aspects of their biology. In this case, instead of vesicle recovery, this is looking at vesicle fusion to the cell membrane. In an even more complicated way compared to light stimulation, electrical stimulation requires the charge and program to travel with the cartridge into the freezing chamber. Due to the energetic aspects of high pressure freezing, it is infeasible to have the sample wired up external to the freezing chamber. So a specially designed middle plate brings the energy needed in terms of a bank of very small capacitors and a very small computer. All of this is triggered by a photosensor bypassing the physical connection that might otherwise be needed. Using a light module, the photosensor on this middle plate is triggered and it carries out the programmed stimulation. This programmed stimulation can be complex or simple, either single ongoing or interval stimulation in order to carry out an experiment. Both of these workflows, light and electrical stimulation, are compatible. It is feasible to both light stimulate a sample as well as electrically stimulate it within the same option. After you have a sample, which is to say it's been preserved by high pressure freezing and it is now a static snapshot of the particular state, there are a wide variety of ways to work with the sample. I've mentioned one of them, freeze substitution, and then going to ultramicrotomy and TEM. I'm going to be looking at a number of these workflows together as they are related techniques, allowing benefit of looking at the revealed layers by ultramicrotomy within the sample. Freeze substitution takes a vitrified cryo sample and brings it back up to room temperature in a process that dissolves the water without it crystallizing, such that you can replace it with resin and turn what is a vitreous sample into a solid resin suitable for sectioning on an ultramicrotome, and then collecting those sections and going to a TEM. Simply cutting, sectioning, staining, imaging can provide enormous insight into what is happening within a sample. But from that process, there are ways of expanding that workflow that I consider worth special note in the context of this presentation. The first is immunostaining. This requires the preservation of structure, often all the way down to protein structure, and allows antibody labeling, either with gold tags by itself or with silver amplification, in order to look at single areas of interest by antibody labeling on an electron microscope. In this particular sample, a knockout mouse is immunolabeled at CX43, and then the gold nanoparticles attached to that antibody undergo silver amplification to make it easier to image. Immunostaining can enable picking out individual areas of interest. Fluorescent labeling is a much easier process in many respects. With fluorescent information that is cross-referenced to the electron microscopy analysis, can take the best of both worlds in many respects. While fluorescent labeling is easier in a lot of ways, and also more common, preserving fluorescence through embedding is often difficult, hence the benefit of imaging before high-pressure freezing and resin embedding. But there are established protocols for this process to preserve fluorescence all the way through resin embedding, allowing direct imaging of that fluorescence within the resin embedded sample. This particular image is looking at a HEC293 T cell with fluorescently labeled EphA2 with M Venus. You can see the fluorescence surrounding the nucleus, as well as a particular area blown up in greater image, and the localization to different parts of the internal structure of the cell. There are a couple different ways of working with a vitrified sample under continuing cryo conditions. Cryofib and freeze fracture are both ways of maintaining the sample under cryo conditions and looking at the internal structure. Freeze fracture is the process of vitrifying a sample, taking the specially designed carriers and opening them up such that the internals of the sample is exposed. In a vitreous preserved sample, when it is mechanically separated, it tends to fracture along lipid bilayers, allowing the direct imaging of proteins crossing lipid layers. In this case, this image shows freeze-fractured intestinal cells presenting very clear images of microvilli at the brush border, as well as some images of mitochondria and plasma membrane between two adjacent cells. Freeze fracture is decades old at this point, but remains a very well established method of looking at the internals of a cell and their interfaces. A very similar technique to freeze fracture is using a focused ion beam to mill away part of a sample. This also exposes the internal structure. When imaging a sample, light can penetrate very effectively through transparent tissue. After high pressure freezing, seeing the inside of it is difficult. The sample needs to be exposed. Freeze fracture, ultramicrotomy, these techniques remove surface of the sample in the general area. In contrast, with correlative techniques or other information, knowledge of where precisely in a sample a particular region of interest is located allows the use of a fib to remove surrounding material, exposing a thin lamella of the area of interest. In the set of images on the right, fluorescence information in the top figure shows some areas of interest in a C. elegans worm. Portions of that worm are imaged in detail in B. After freezing, that area can be examined by electron microscopy. In D, that area can be exposed with the fib beam. Subsequently, E through H shows lift out in which the lamella is removed and presented for TEM imaging. The lower figure shows that imaging. In this sample, the areas of interest were the ribosomes. It is a variety of targets, both looking at individual ribosomal translocation states, as well as some of the cell-to-cell variation in ribosome structure. Overall, the use of a fib enables a great deal of versatility in how the sample is exposed, but it does require targeting. The comparatively very large vitrified volume of a high-pressure frozen sample does mean that fib techniques need to be carefully considered in looking for particular regions of interest. Still, despite that caveat, it can be a very illuminating technique in looking at a particular aspect of interest within an overall sample. The last workflow that I'll be talking about is SEMOVIS. SEMOVIS stands for cryo-electron microscopy of vitreous sections. In a very similar fashion to sectioning of a resin-embedded sample at room temperature, SEMOVIS is the sectioning of a cryo sample at cryo temperatures, maintaining it in its native state without any substitution. Cryo-ultramicrotomy is both a difficult and rewarding technique that can allow you to image a sample without any chemical modification. The sample is preserved in high-pressure freezing, sectioned, and then those sections are imaged. In this particular image of gram-negative bacteria, this is what the bacteria looks like, and that's it. The peptide dog-glycan layer is shown, as well as the plasma membrane. These sections, these vitreous sections, are quite fragile, but with skill, they show the sample in a fully native state, simply vitrified to capture that particular moment. In examining all of these workflows, they work not all at once, but they come together to offer a large variety of options to go through the EMIs and end up where the sample is best suited for subsequent imaging. Starting with live cells, carrying out any appropriate imaging, high pressure freezing on the EMIs, any light or electrical stimulation on those images as appropriate, and then taking that vitrified sample and proceeding through the variety of cryo or room temperature workflows to end up in an electron microscope and collect the appropriate information from the goals of the imaging. Thank you for listening to my presentation. I hope this was informative and useful. Please reach out to Leica Microsystems if we can help further in your process. Thank you for your time.
Thank you, Seth, for your informative presentation. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, why are different types of carriers used?
Well, generally a couple of reasons. One kind of historical, just as the volume for which high pressure freezing could be successfully implemented expanded, so did the volume of the carriers to work with larger samples. In any particular moment, now or in the past, the shape of the carrier used generally was chosen based on how to work with the samples. In Samovis, for example, copper tube freezing is a particular way of presenting a sample that is a small volume, but useful for sectioning. Whereas in freeze fracture, a more spherical sample is often beneficial. In all cases, the physics of high pressure freezing remains unchanged. It's just how the sample is presented and handled that affects the shape chosen.
Great, thank you. Our next question asks, are sapphire discs only necessary for light microscopy workflows?
Necessary is a bit of a strong word. Sapphire discs are necessary for light microscopy workflows. They are occasionally very beneficial in unusual circumstances in other workflows. Generally for cells that are just very delicate or very contrary, and despite any coatings on the metal carriers, the cells in question refuse to grow. The inert material of the sapphire disc is sometimes a last resort, despite its greater cost for getting cells to behave and grow more normally.
Great. Our next question asks, how does the ice switch between these different workflows?
So the cartridge system means that any particular carrier shape can be done without any changes to the ice. You could do a freeze fracture system, a sapphire disc, CLEM experiment, or copper tube freezing, or regular carrier freezing, all in the same session. So if it is only a question of how the sample is fed into the ice, generally speaking, you can swap basically at will. For the light stimulation and electrical stimulation, there is some hardware changes which take swapping of, about 3 to 10 minutes. It's very short for the light modules, so just remove and swap a plug on the front of the machine. The electrical stimulation uses a small plate in the front, which takes about 5 or 10 minutes to swap out. The sampling chambers use an involved apparatus that is longer to install on Leica's service side, but still relatively straightforward to swap out. It takes also about 10 minutes, so it would not be appropriate trying to do any kind of live cell rapid timing workflow in part of other samples, but you can swap between them in the same day back and forth quite easily.
Thank you, Seth. Our next question is, how many samples can it do?
Well, for any particular day, I would say that the limitations have, in my experience, always been on how many samples can you prepare. So the ice can freeze a sample roughly every 2 minutes. So you can freeze quite a lot in any single session. The ice is designed to freeze in 3 groups of triplicates. So 9 samples in a batch. And you can do as many as you have in that time rate. For the overall lifespan of the machine, it is recommended that you get it serviced about every 1,500 shots of freezings of a sample. And that takes most users I've worked with a couple years or more to get to. Generally, batches are around 9 to 27 samples in a session.
Great, thank you. Our next question asks, using this system, do you see any crystalline ice, or is it all vitrified? What kind of pressures do you use to achieve this state? And do you use any cryptoprotective agents, such as DMSO, during cooling, or just cool as it comes?
Okay, a few different questions in there. The answer to all of them kind of at once is yes. The pressures that we use that we work in with high pressure freezing is around 2200 bar. There are some very carefully done studies suggesting that the ideal range is between 2000 and 2400 bar, for which the particular cell type can cause some shifting in the ideal pressure. 2200 bar is the most common successful pressure for the samples that I've observed. Now, that ties into the other part about, is the sample entirely vitrified? Generally speaking, it is dependent on how thick you're trying to work the sample into the ice. The depth of freezing is usually the main factor in initial tests of most samples. So you might see good freezing for a cell monolayer, 100 microns depth, great freezing all the way through it. If you used deeper carriers, say 300 microns, you would very likely in that same sample see great freezing at the surface and then progressively lower quality freezing as you get deeper into it. For looking at tissues and such, it can be very important to get good vitrification all the way through the full depth of the tissue. And so cryoprotective agents are often used, and they vary based on how the tissue would be physiologically affected by the cryoprotectant. For example, when I was just starting out, I came across a reference. High molarity sugar water is a great cryoprotectant. It is very hard to crystallize that water did high pressure freezing, and you will not get any crystalline ice in that water part of it. After I tried it a couple times and then went back to the library, I found out, perhaps very obviously to many people in the audience, very high concentrations of sugar will completely mess up the cells in the sample. So cryoprotectants such as DMSO, 2-methylpentane, and others are all chosen based on ensuring the sample will not be affected. A general rule of thumb, most broadly speaking, is the less water you have surrounding the sample, the better off you'll be for avoiding water crystals. I hesitate to make a direct recommendation of any particular cryoprotectant. performance of them is often dependent on the sample. And so usually, as I've experienced myself, some time in the library will give a better recommendation than trying to experiment with the full breadth of possible options.
Thank you. It looks like we have one more question here, and it says, I have a number of colleagues that are interested in high pressure freezing, and I would like to share this presentation with them. Is there a recording available for sharing?
Yes, this is an on-demand webinar, so the link to this page will let anybody view it again. If there is an interest in the slides of this presentation, I'm afraid you would have to directly reach out to me. This is something we will not be distributing on demand, though it will be available through discussion, is probably the best way to put it.
Yes, thank you, Seth, for answering those questions for our audience today. Do you have any final comments?
By all means, reach out to me at my email address. It's seth.villarreal@leica-microsystems.com. I'll put that in the next version of this presentation written somewhere. And thank you for your attention.
Yes, thank you again, Seth, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.